Hello everyone, good morning. I am Dr. Prisilla from Biotechnology Department. I am going to explain about the topic of protein types and structures. What is protein first of all? Protein, it is made up of amino acid. It is a macromolecule. You can see here it is polymer of amino acid formed polypeptide chains. It is play an important role in various living organisms. It is acted on hormones, enzymes. It is also acted as a structural element in their environment. Coming to the different types of protein configurations. So protein, it is basically structural organization. It is categorized 4 types. First one is primary structure and secondary structure of protein, tertiary structure of protein and quaternary structure of proteins. And coming to the discussion about primary structure of proteins. The primary structure of protein, it is very linear, simple in structure. We can easily determine the genetic diseases and also we can easily refer the numbers and sequence of amino acids, and along with the amino acid are attached with each other through the peptide bond. It is also determined with the help of end group analysis, Edman analysis and sequence analysis. These are all the points will be important for protein structure and primary structure of proteins. And next is secondary structure of proteins. It is also having different types: alpha helix, beta sheets, beta bend, beta turn and random coiling, random loop, ribbon, stick, and so many models are there. But based on the alpha helical and beta sheets conformations, it will consider as a secondary structures of proteins. So the linear chains of protein, it is undergo the protein folding due to the presence of the hydrogen bond, which is result in the formations of helical structure. So that's why that helical structure has formed based on the polypeptide of hydrogen bonding. It can may either intramolecular or intermolecular structure in the proteins. And along with the protein classified, there are so many types are there. Here it is based on the structure, it is classified 2 different types. One is alpha helical structure, another one is beta sheets. And coming to the discuss about alpha helical structure. And alpha helical structure is, of course, you know that the alpha helix secondary structure of protein, it has a more number of hydrogen bond between amino group and carboxylic group that will be stabilized in the structure of alpha helix, and followed by, you can see here, the inter and intramolecular. As I told you before that slide, here I am just highlighting the intramolecular hydrogen bond that will be present in the alpha helical structure of secondary structure of proteins. And it is also having 3.6 amino acid per turn in each turn of helical structure. And helix, it also can right-hander or left-hander in their nature. That protein folding, it will be happened. Okay. And next is alpha helix can be formed by either D amino acid or else in L amino acids. And mostly alpha helix of amino acid, alpha helix of protein, it is acted as a transmembrane protein for all of any globular proteins. So these are all the informations related about alpha helix structure of proteins. And next is followed by, we can see the next types of secondary structures of protein, that is nothing but it is a alpha helix structure. Here I am just highlighting the picturized images which will be taken from a protein data bank. It is first image, it is clearly note down the ribbon like structures of alpha helix, and next image it is a loop and stick model of alpha helix structures of proteins, and next is space filling protein structure. So these are all the some different image highlighted for alpha helix structures of secondary proteins. And next we can see about the beta sheets. So beta sheets is there in the intermolecular. As I told you before that alpha helix, where it is present in the intramolecular. So here I am just highlighting the differentiation between the structural organizations of secondary structure of proteins. So the beta sheets, intermolecular hydrogen bond that will be present in carboxylic and amine group of amino acid, which is converted into the next, followed by that will be adjust in the adjacent amino acid, and more than one linear change will be present in their overall structures of alpha and beta sheets of proteins. And Here I am just highlighting what are the amino acids are present in the beta sheets. The glycine and alanine will play an important role in the formations of beta sheets. So that here we can confirm that beta sheets how it is formed based on these two amino acid residues. And next type is random coiling. Random coiling, it is a presence of adjacent isoleucine, that is charged amino acid. It is lead to the formations of secondary structures of random coiling loop formations in their particular nature. And next is gamma helix and beta sheets of secondary structures of protein. So gamma helix, it is also characterized by the high number of hydrogen bond is present in the gamma helix structures of secondary structure of proteins. And beta band, beta turn or hairpin band, they are also discussed with the some amino acids are present. Here I am just highlighting for proline and glycine, that is causes the reversal of protein, that is directed through the formations of beta bend. So that is why we call it as a beta bend or beta turn. So here I am just highlighting the tertiary structures of protein, how it does look like. So once we can take it the primary structure, that is very simple in nature, we can easy to analyze the sequence present in the primary structure, and followed by here the extended structures of secondary structure, and it will somewhat complicated in the structures of tertiary structure. I am just highlighting for the hemoglobin in our blood, and followed by quaternary structure. It has a 4 segment, 4 different types of hemoglobin is there, that is linked with the polypeptide chains. And along with the some more important point will be noted for tertiary structures of protein. It is also interaction between amino acid that are present far apart, and then far apart of amino acid that will each other to leads the formations of tertiary structure of protein. And followed by we can see the where that interaction will be there, how it is connected. So here I am just highlighting how that protein will be connected to form a polypeptide chain. The hydrogen bond, hydrophobic bond, disulfide and ionic bond, that interaction will happen where in the protein structure, tertiary structure of protein are present. And one of the good example of tertiary structure is myoglobin. So the myoglobin is an oxygen binding protein. It is present in our muscle cells. So that the muscle cells are having that oxygen binding protein. So it is also contains 153 amino acid in single chains of polypeptide. So that it is characterized by presence of 8 helical segment that will be joined together in the only one linear chains of amino acids. And here I am just highlighting the myoglobin structure. As I told you before that slide, it is 8 different types. So here, so for A to H, that is a different types of myoglobin and combined together only one single polypeptide chains. And next is of course that myoglobin are having the 2 amino group and carboxylic group, and along with polar and non-polar amino acids. Here I am highlighting polar amino acid that will be present in the external surface of the area, and non-polar amino acid present in the internal structures or internal structure that will be forms a hydrophobic environment in that area. And R group is, of course we know that R is nothing but the side chain of amino acid. The R group of amino acid, it is present in E and F type of hemoglobin helical structure that will involve in the interactions with heme group to carry and binding with the oxygen through the muscle cells. And iron atoms has 2 coordinations bond is there, one with histidine amino acid and other with present in E helix and other with the amino acid. This is a orientation, structural orientations about the myoglobin. And bond of oxygen, it is stabilized through the interactions with the histidine amino acids. So these are all the some important points which will be noted for the tertiary structure of protein. And finally, next structure, that is a quaternary structure of protein, that is 2 or more. This polymer of amino acid to form a polypeptide chains in their complex in nature. So proteins which 2 or more polypeptide chain that will be interacted each other, or within that polypeptide chain how it is interacting, and through hydrogen bond or hydrophobic, ionic and disulfide bond, it will interact each other to form a quaternary structure of protein. And Next is each polypeptide it will be referred to as a subunit. That subunit will be easily identified with the numbers of hemoglobin. So these are all the different types of polypeptide chains, how it is noted down the quaternary structure, and along with the subunit it can be same, it will be vary based on the environment. And good examples of quaternary structures of protein, it is hemoglobin. So hemoglobin is also having 4 polypeptide chains, and here I am just highlighting the hemoglobin A because there are number of hemoglobin types are there in our body. So hemoglobin A has 2 different chains, one is 2 alpha chains and 2 beta chains in their structure. Each chain has binding sites for heme groups that will be connected with the polypeptide chains. And here I am highlighting the 3-dimensional structure of hemoglobin. It will be taken from the PDP protein data bank, it will be retrieved. We can easily retrieve other protein also because of hemoglobin quaternary structure. I am highlighting the structure, how that orientations are formed, how it is linked with other types of hemoglobin group to form a polypeptide chain in only one complex in structure. So this is a complex structure, so it is based on the complications and orientation organizations of quaternary structure of protein. It is comes under the category of quaternary structure of protein. Thank you all.